Alright, so this is a quick video describing what happens when we have mutations to our DNA. We've been talking about transcription and translation. So anytime we have a change in our DNA, we get a mutation. That's actually the term we use for changes of DNA. And it is the source of all variation amongst organisms, is due to these mutations. Now there are a couple of different kinds of mutation. So the first type of mutations are actually at the chromosomal level. So in mitosis, we talked about how the DNA condenses down into chromosomes. Well, if we have mutations that occur at the chromosomal level, we can have a couple of things happen. So the first one that can happen is if we have a deletion. If we have an entire chunk of a chromosome taken out, we are going to actually lose whole genes. We can have duplication. So if during DNA replication, if the DNA gets broken and they make more than one copy of a gene, so if we look right down here, using the letters as our shorthand for the genes, we've added an extra B gene, and that's a duplication. Our next one is an inversion. Now inversions can be a little trickier. It's basically when we have all of the same pieces but they're not in the right order. This can be a problem when our enzymes are signaled to make a copy of a gene at a certain location, but what they're making a copy of is not the gene that they were anticipating. Our next one is a translocation. Now a translocation is actually taking a gene, not just switching what order it is on the same chromosome, but actually taking a gene from a completely different chromosome and plugging it in. And again, enzymes have a really hard time locating these genes that they're supposed to be working with if it has been translocated. Now the other set of mutations that we have are gene mutations. Now these are ones that are a little bit closer to what we've been working on currently with the transcription and translation. So if we have a mess up in the DNA or a mess up in transcription, so our mRNA has the wrong base, we have only one gene being affected and it changes that actual sequence. It changes what's being made. So we have our point mutations. Our point mutations change one single base. One thing is different, that's it. It's also called a substitution. So for example, if I went from CAC to CCC and that A changed to a C, that could be either silent if our amino acid that it codes for doesn't change. So if we're making the same thing anyways, we consider that a silent mutation. It could be a missense mutation, so we're making basically a mistake in the sense of what we're making, where the amino acid is coded for as something else. So we're not making exactly the same thing. Or we can make nonsense, and we're basically not going to finish making the protein, and that's where the amino acid it gets coded for is a stop codon, and it makes a really short nothing of a protein. Now the other thing that can happen, if you'll recall, we read our mRNA in codons or sets of 3. So we also have our frameshift mutations. Now for the frameshift mutations, if I add in one extra nucleotide, we actually shift and we will read. So for example, here we have a CAC. If I add an extra A in here, I now am going to read CAA, and this C off at the end, that's going to end up being the first letter of the next codon. And so it's going to mess up every single codon after that base was added. The other thing that can mess it up is a deletion. If we went from CAC and we took out the A, we only have CC, it's going to take that first nucleotide from the next codon, and again it's going to mess up every single amino acid after the mutation. That's basically our main 5 mutations that we're going to be looking at in the genetic as far as our gene mutations, and this was our mutations notes. Thank you.